Hi, everyone. Thank you for attending this session of the Brain Science Speaker Series presented by LSU's Multidisciplinary Initiative for Neuroscience Discovery. LSU Mind aims to coordinate and strengthen neuroscience research, education, and community outreach here at LSU. I'm Susan Duncan, and I serve on the Executive Committee for LSU Mind along with Melissa Beck, Neela Donovan, and Heather Lucas. We would like to thank the LSU Center for Computation and Technology for funding and supporting the Brain Science Speaker Series, and we'll be hosting additional speakers and events in the next academic year, so please stay tuned to the LSU Mind website and sign up on our mailing list. So today, for Mind's second ever virtual event, we are extremely excited to have Dr. Yasmin Hurd presenting. Dr. Hurd is the director of the Addiction Institute within the Mount Sinai Behavioral Health System, as well as the Ward Coleman Chair of Translational Neuroscience and Professor of Psychiatry and Neuroscience at the Icahn School of Medicine at Mount Sinai, New York. Dr. Hurd is an internationally renowned neuroscientist whose translational research examines neurobiology of drug abuse and related psychiatric disorders. Her research exploring the neurobiological effects of cannabis and heroin has significantly shaped the field. And using multidisciplinary research approaches, her research has provided unique insights into the impact of developmental cannabis exposure and epigenetic mechanisms underlying the drug's protracted effects into adulthood and even across generations. Dr. Hurd's basic science research is complemented by clinical laboratory investigations evaluating the therapeutic potential of novel science-based strategies for the treatment of opioid addiction and related psychiatric disorders. And based on these high-impact accomplishments and her advocacy of drug addiction, education, and health, Dr. Hurd was inducted into the National Academy of Medicine that complements other honors she's received in the field. We are very excited that Dr. Hurd has been able to join us today, including a visit earlier with students and trainees to allow them to ask questions about her interdisciplinary research and the path to her current position. So please join me in welcoming Dr. Hurd now for her talk entitled The Vulnerable Brain, Pathways to and from Addiction. Thank you, Dr. Hurd.
Thank you very much, Susan, for the invitation. And actually, it was really wonderful meeting with the candidates earlier today. And I think you have a really, if they're representative of your trainees, I think you have a great group. So it was really wonderful. So when I was asked to give the talk in one way, it was to provide a more broad stroke overview of my field, which is the neurobiology of addiction. And an important aspect of that is, understanding what we know about the paths that may lead to a brain that makes one more vulnerable to addiction, but the information that we have gained, how that can help us start developing new treatment strategies out of addiction. There's not much time to go. I will tell you, I'm going to be biased. There's not as much time to talk about all of our great work in terms of where we're going in novel treatments, but I'll end with one of them and can, in the discussion time, talk about a few others. So here we go. So we know that for me, the US, I say, is a country of addiction. There are over 30 million people with a substance use disorder, and that doesn't cover people who also misuse the wide classes of drugs that people consume. I mean, a huge part of our issue with substance use disorder, I think, is the economic, is not just the cost to the patients and their families and communities, the economic that combines with everything that makes it for a huge economic cost. And a lot of this is really linked to in part, the criminal justice system, for which I don't think that substance use disorders really should be in there. But it's a health issue, and a health issue that's compounded by the fact that there is such a stigma with substance use disorder, and the overdoses, obviously, that are a huge part of the past decade. But all of these things together has really put the healthcare system under siege. And I don't think that, the average person realizes the degree to which substance use disorders are a major health impact that we need to understand on that level, and not some aspect of a criminal justice that we need to focus on. The health is really critical. And especially this past year with COVID, where the social isolation has actually increased drug use in our country, where these are just looking at some of the national increase in, this is just unbiased looking at urine tests, and you see like opioids are absolutely higher, and as well as stimulants. But one thing that is missed in our horrible pandemic is the fact that we still have an opioid epidemic of overdoses. So over 85,000 people died within the past year from drug overdoses, the majority of those still opioids. So we can't let go of that as we fight the other issues that we have going on in our society today. But the fact that addiction is a huge public health crisis. So what we've studied the neurobiology of addiction for a very long time. Probably we needed to study, obviously there's a lot of nuances to the neurobiological mechanisms that we're learning, but there's some fundamental things that we do know about the neurobiology of addiction. I always say it's a whole brain disorder. And yes, we know that there are certain regions of the brain that underlie certain behaviors relevant to addiction. For example, the nucleus accumbens that many people will hear about, that ventral striatum that's important for reward, expectation, goal-directed behavior, parts of the dorsal striatum that's important for habit formation, or the prefrontal cortical areas for cognitive control, cognitive flexibility, motivational drive, or the amygdala, this region in the temporal cortex that's really important for emotional regulation, drug-seeking behavior, as well as like the hippocampus for memory. So we do know some fundamental things about the neurobiology in that context. And we know that many substances of abuse, whether it's alcohol, opioids, stimulants, cannabinoids, they impact on the transmission of neurotransmitters, especially, for example, dopamine, but there are a host of other transmitter systems that are really critical for substance use disorders. And I'm going to, talk about a number of them along the way. But I think that many people, even if you're not in this field, always think of addiction through the lens of like dopamine. And because dopamine is that transmitter of reward, for example, and so when people use a drug, especially those that are highly addictive, dopamine levels go up, there's euphoria, and when dopamine levels go down, there's this negative state, this dysphoria. We also know aspects of, like I said, the brain regions that are activated from human imaging studies, for example, and a host of animal models where the nucleus accumbens, this ventral striatal region, activated when people, for example, will experience a drug. But as the addiction process goes, so we talk about, addiction is not just one stage, and addiction is definitely not just, is not taking a drug. Taking a drug and the intoxication with the use of that drug is not addiction. What we see in the process of the neurobiological changes that occur over time and the different stages of the addiction cycle are aspects of, of the synapse, the synaptic communication. And a lot of this is regulated by the transmitter glutamate, the excitatory transmitter that we see in terms of from the prefrontal cortical areas. And of course, I'll talk about other transmitters like GABA and endocannabinoids as well. But in the context that it's synaptic plasticity that is impacted, not just dopamine over time. So a major question for many people is, why is it that many people might experiment with a drug that's highly addictive, but only a few go on to have a problem? And that has been, I think, one aspect where there is stigma with substance use disorders, because many people think, oh, it's just that person has low morals, or they're weak and so on. But there's a lot to it that I think that many people don't understand relevant to the neurobiology of addiction. And so I'm going to go over, like I said, big strokes of what are some key important aspects that are relevant to neurobiology. So addiction risk covers to me a number of different things. We know that neurodevelopment does play an important role in laying the foundation for the sensitivity that make someone perhaps more vulnerable to addiction. The environment along that developmental process is key. So aspects of, stress, obviously early drug exposure. On top of that, you have genetics. And when the genetics interact with the environment, we know that there are these epigenetic interactions that, again, can change how the brain responds to a drug that it might see later on in life. And that combination impacts on behavioral traits that also lends itself to addiction risk, whether it's reward sensitivity, deficit, inhibitory control, impulsivity, and anxiety, depression phenotypes. But of course, all of this is moot if you don't take the drug, even if you have all of these developmental, genetic, environmental, all of these aspects. And so the drug use does matter for that. So I'm just going to go over, like I said, a couple of broad strokes in, aspects relevant to this individual vulnerability to substance use disorders. So if we take genetics, for example, we know that the risk for becoming dependent on any drug after sampling it is, each drug has a different impact on neurobiological systems, even if they may converge on, for example, like I said, dopaminergic system. But for example, nicotine is highly addictive, and so is heroin. And on top of that, you have the heritability that feeds into aspects of genetics. And for some drugs, like psychostimulants, like for example, cocaine or opioids, you have a high heritability for drug addiction. And if we take the opioid, if we take, for example, heroin, we know that opioid abuse has a pretty moderate to high genetic load. So there's about a 40 to 60% vulnerability for genetic risk. And we can, for example, look at aspects of our own endogenous opioid system to see whether or not genetic influences there might contribute to some of that vulnerability for opioid addiction. So our endogenous opioids, we have our endogenous heroin, for example, beta endorphin, these peptides and enkephalin, and they stimulate the receptors that mediate reward and positive mood, like the mu-opioid receptor. We also, I'm not going to talk about it today, we also have the counterbalancing opiate peptide system, like the dynorphins, kappas, that mediate negative mood and dysphoria. So we have now known for a number of years that genetic polymorphism of receptors that mediate, perhaps, or I should say the hypothesis was for many years that a genetic polymorphisms of the receptors that mediate the pharmacological actions of drugs. So in this case, the mu-opioid receptor for opioid drugs, like heroin and morphine, that if those genetic variants are functional, they could perhaps change the sensitivity to when someone takes that drug. So one of the first genetic clues that this might happen in regard to opioid addiction was this functional polymorphism of the mu-opioid receptor. It's called this. I'm not going to go into the variant, but it was known from basic science research using cell cultures that it changed the functional activity of how the receptor functioned. And then people started looking at clinical studies. And we looked in our subjects who had, this is a postmortem, died from a opiate heroin overdose. And we could see that those individuals who had this genetic variant of the mu-opioid receptor, nearly 90% of them, over 90% of them, were heroin users in our population. So clearly there must be something of a genetic risk, either not just for developing an addiction, but perhaps even the overdose that came with it. When I went to Mount Sinai, one of the things we did was to even take people who came through the emergency room who were having drug overdoses, and we could see again that genetic polymorphism of the mu-opioid receptor actually predicted the clinical severity of individual outcomes with drug overdose just from the emergency room. Since I'm a neuroscientist, the thing for us is trying to understand when we see aspects of vulnerability to see what does it mean for the brain on a functional level. So opioids bind to this mu receptor that then initiate a cascade of events through these signaling mechanisms. And one of the things that we could see that indeed, people who have polymorphism, those specific functional polymorphism of their mu receptor, their signaling cascade differed to when an opioid would stimulate that mu receptor. So, and the downstream signaling, here's just a gene called L, the transcription factors that mediate the cellular responses that occurs in following stimulation of the receptor. So you have the genetic polymorphism. It's not all polymorphism obviously are functional, but they're polymorphism of mu receptor that are absolutely functional, so that any two persons taking the same opioid drug will their cells respond differently. Obviously, genetics also plays on behavioral trait and psychiatric comorbidities. And we know that there are a number of behavioral traits that, as I said earlier, that links to aspects of addiction risk. So when I'll use cannabis use disorder for one of this, because many people think that you can't actually develop addiction to cannabis, and that's actually quite wrong. In fact, one of the largest groups in the country that have that with a substance use disorder, it's for cannabis use disorder. And we know that many people who use cannabis, about 30% of them will develop a cannabis use disorder in their lifetime. So that is, it's really concerning. But one of the things that we see that with cannabis use disorder, there is a high psychiatric comorbidity. For example, there's a high degree of anxiety disorders and a schizotypical type of personality disorders. And when you look in the brains of people, and for example, neuroimaging techniques, looking at people with a cannabis use disorder, a lot of the neurobiological changes that are seen are actually very similar to what you see with other substance use drugs that are abused. So for example, you have changes in the amygdala, you have changes in this orbital frontal cortex region that mediates, again, goal-directed behavior, decision-making. So we know that it's not a unique drug in regard to just what we see that's changed in the brain. But one of the things, in coming back to like behavioral traits of vulnerability, because many people in our society will experiment and use cannabis, and now even more than ever, but not everybody goes on to have a problem. When we look at behavioral traits of those individuals with cannabis use disorder, one of the things that's clear is that many people have much greater anxiety and neuroticism traits. There's also higher aggression traits, and the negative affective traits we see in many different measures when we bring people into a lab and look at their behavioral traits. We can even combine that with genetics, because as I said, why is it that so many people will experiment with cannabis, but not everybody goes on to develop a cannabis use disorder? And when we look at genetic variants that we can see have a functional level, at least in the human brain on a molecular level, that having this variance may change the expression of either of different transmitter systems relevant to addiction, we could see, for example, that if you have a high anxiety trait, your risk may increase a certain amount. If you have the genetic risk, you might increase your odds for cannabis use even greater. But that combination of having the high-risk genotype and a high anxiety neuroticism trait could increase your risk for cannabis dependence even 9-fold. So everyone is not the same when they consume cannabis. There are a number of issues goes into who becomes more at risk. So what about the environment? As I said, we know that environment makes a really big difference. And doesn't matter the socioeconomic, the racial group, the earlier initiation of drug use, or I should say, early childhood abuse leads to earlier initiation of drug use, whether it's cannabis, cocaine, or heroin. So early life trauma is very critical for increasing addiction and actually psychiatric risk. We also see, and this is from Karen Baki at our group at Mount Sinai, where looking at neuroimaging in adults, and these are adult individuals with either controls or have cocaine use disorder, and you can see that the structure of their orbital frontal cortex is reduced in those individuals that had the higher childhood trauma. So this is a long-term impact on the brain in brain areas relevant to addiction vulnerability, relevant to decision-making and cognitive flexibility. And it also correlated to the depression, depression traits that these individuals showed. So early life trauma during those negative environmental, I should say, those negative environments that have people are raised does have a long-term impact. One aspect for that, for me, that was always intriguing was, when we look at the adult brain of people who have a substance use disorder, substance use disorders are not single gene disorders. Absolutely not. But there's a characteristic that many people who were doing neuroimaging studies had seen, that this low dopamine D2, and dopamine, the D2 receptor, obviously mediates the action of the dopamine transmitter. And when you looked in the striatum of a lot of these individuals who had a substance use disorder, whether it's psychostimulants or opioids or alcohol, and even cannabis, one of the things that's characteristic is this low level of dopamine D2 receptors. There's always the debates in the field of the chicken and the egg. Is it drugs that induce this reduction of dopamine D2? And yes, they do. Animal studies show that. Or was it there to start? And there's also evidence that individuals who might have a lower D2 to start may be more at risk. But coming back to the environment, there was a great study that Mike Nader and Morgan did many years ago, and that I think set a foundation that really, again, opened people's eyes to that the social environment makes a difference. So these are monkeys that were individually housed. And you can see that when they were then socially housed, this is their dopamine D2, I should say, in the striatum, you can see it. And when they're socially housed, their D2 goes up. I think these studies actually have really important implications for today when we think about, COVID, and where most of us were socially isolated for a year, and you can see that dopamine D2 levels are lower during that time. So perhaps that might even contribute to the greater substance use that we see during this time. But when they're socially housed, their dopamine D2 went up, but not in all animals. Interestingly, it was lower in those animals who later on, it turned out, were subordinate, because during social housing, certain animals became dominant and others became subordinate. And living in a social environment where you're subordinate, your dopamine D2 levels are lower. And those are the animals also that self-administered more cocaine. So social environment that we can see from animal models and human imaging studies and animal imaging studies makes a big difference to a lot of neurobiological marks that we know are linked to addiction vulnerability. Another area I was just looking to make sure I keep the clock going, because with Zoom I can't see the time. But another area about vulnerability and individual risk, I started mentioning with the environment and neurodevelopment. And neurodevelopment is really key, that I think people don't appreciate enough. And not just for addiction disorders, but for most psychiatric disorders, we know that even though they are expressed during adulthood, the genesis, they actually start during early development. Here, I'm going to focus a little on cannabinoids, and mainly to emphasize two things. So when we look at vulnerability in terms of who, we have an adult population of heroin users, and we looked at genetics. When you look at, some aspects of what people did earlier in their lives, cannabis always comes up. but the endocannabinoid system, at least during, plays a really critical role during most of the neurodevelopmental processes, from early in fetal development, from neurogenesis, migration of neurons and glia, to how cells communicate in terms of synaptogenesis, and well into adulthood with synaptic pruning, and that final last part of the maturation of the prefrontal cortex. The endocannabinoid system plays a really critical role. And there have been a number of longitudinal and epidemiological studies that have shown that early exposures to cannabis during this neurodevelopmental processes can lead to, many different negative children outcome, and even in young adults, you see aspects of psychosis, anxiety, and so on, and even leading to cannabis use disorder. So one of the focus of our research is on the developmental effects of cannabis. And today I call it the perfect storm. And in part, we're in a perfect storm today because of the wide availability of cannabis and cannabis products now due to the legalization and decriminalization of cannabis for medicinal and recreational use in many states. And therefore, this reduced perception that cannabis is of any risk. And on top of that, we have increased THC concentrations than ever before. So together with many other factors, to me, it's a perfect storm for the vulnerable developing brain. And we can look at cannabis exposure from the prenatal time period and different developmental times as well. So when we look at the endocannabinoid system in the brain, and this is looking at cannabinoid receptors where cannabis binds, but this is looking at the genes that express these cannabinoid receptors. In the adult brain, the cannabinoid receptor is actually the most abundant G protein coupled receptor in the brain, and G proteins are the cell surface receptors. It's highly enriched throughout the cortex, throughout the dorsal striatum in particular, the amygdala for emotional regulation, the hippocampus, like memory, and the cerebellum, motor coordination. But when we look at the brains of fetuses, we can see that the cannabinoid receptor has a much more restricted expression, especially during this mid-gestational period, to areas, for example, the nucleus accumbens, which is this ventral part of the striatum, really, really robust in the amygdala and in the hippocampus. So when we look at the brains of fetuses, and as I mentioned before, the endocannabinoid system is critical for many neurodevelopmental processes, and during the fetal time period, even for hardwiring of the brain in terms of the axonal tracks that are made. And when we look at the brains of fetuses whose mothers smoked cannabis, we could see in many different ways in which we have studied it. For example, there are changes in these microtubule binding proteins, which make up the cytoskeletal, provide cytoskeletal stability. So this is, for example, statin 2. There's this reduction that we can see not only in our animal models, but in the human fetuses, and even in multiple brain areas. But if we look at, even in a biased manner, a hypothesis-driven manner, the dopamine D2 receptor that I mentioned earlier is a classic characteristic that's seen in adults with substance use disorder, this reduction. You can see that these fetuses have a reduced expression of dopamine D2 even during this early stage of fetal development. We can also see it in the amygdala, the brain region really important for emotional regulation. And here it emphasizes one thing, like there's a sex difference. Already during fetal development, you see differences that here it's males that are much more sensitive to their mother's cannabis use, at least in terms of their amygdala cells are more sensitive. So in human subjects, people will obviously consume other drugs than cannabis. And so the specificity of what are the neurobiological changes that we're seeing, and it's always something that people may question. And also, for me, a major question for our line of research has always been, what are the long-term consequences later in life, and especially on the adult brain? And so that's where our animal models come in, and we can actually replicate a number of the molecular changes that we see in the human brain in the animal models when we give them THC. So we know that THC is mediating these changes. So for example, here is the dopamine D2 receptor. Again, we see this reduction in their nucleus accumbens, and it lasts into adulthood, even though they never see THC again. We can see changes in the firing activity of their cells and the morphology of their cells and so on. But what about behavior? So that's where we had looked at, it's not necessarily we're looking at the gateway hypothesis, but we wanted to see, does early exposure to cannabis or THC here, does it change the sensitivity to opioid drugs? And here we allow animals to self-administer heroin. And we could see that those adult animals from our earliest studies, I should say, that those adult animals with prenatal THC exposure, they self-administered more heroin. But interestingly, even when we made the conditions so that overall they self-administered the same amount of heroin, those animals that had had prenatal THC, they run to that to get that first hit of heroin. And importantly, they self-administer more heroin during stressful conditions, even mildly stressful conditions. So we know that prenatal THC has a long-term impact on adult behavior relevant to addiction or opioid sensitivity. For me, I always, a lot of our research comes back to the relevance to the human condition. And of course, it's very challenging to do longitudinal studies in humans looking at the impact of fetal experiences on their behavior later in life. I'm masochistic, and one of my colleagues, Yoka Nomura, is more masochistic, and Yoka has been studying the prenatal effects of stress. And so we combine that in looking at prenatal effects also of cannabis. And we decided to look at the placenta because we wanted to get an idea as to the environment, the molecular environment that the fetuses are experiencing with maternal stress or maternal cannabis use. And we could see, interestingly, that there was a significant reorganization of the transcriptome, the thousands of genes that are expressed in the placenta. And for example, just looking at one of them, this is the cannabinoid receptor. It is changed, and it is changed especially in relation to the amount of cannabis that the moms reported using. But we also saw something very surprising, that there was this really strong reorganization of immune-related genes. So the immune dysregulation was a huge part of the placental changes associated with maternal cannabis use. We saw that there's a double hit consequence, that though the changes in the placenta, that where both exposure to cannabis and exposure to stress increased the glucocorticoid, the stress-related genes, and had a more profound impact on the cannabinoid genes. But does that mean anything for their children outcome? So that's where Yoko, we've been doing these longitudinal studies and looking at these kids. And one of the things that's been interesting is that you do see behavioral differences, even from 6 months old, and we've been studying them. And so I'll just show a few of the findings. Once again, double hit matters. I'm showing this first before I focus on cannabis. But for example, anxiety disorder, if they had both cannabis exposure and stress exposure, very high levels of anxiety and aggression. But with cannabis use, we could see, similar to the changes that we saw in the placenta with this high sensitivity to glucocorticoid stress hormone system, in these kids later in life, their stress hormone levels of cortisol is higher. And their clinical behavioral scores, because they're studied every year, their clinical behavioral scores, for example, higher anxiety, higher aggression, higher hyperactivity. So we know, that we see these changes in the placenta and reflecting some aspect of their environmental exposure in utero. We see changes on behavior. And so for us, the question is there anything predictive about the placental changes to behavior? And one of the things that we see is that, yes, there is. those genes that were related to immune dysregulation correlate significantly with the anxiety behavior exclusively in those kids with maternal cannabis use. So we know that maternal cannabis exposure has, like I said, this profound impact even on the placenta, and that it leads to, or at least it's associated with protracted effects on, I didn't show you physiological measures, but we've done those, and we can, other than court, and behaviors. And some of the behaviors that we see are actually behaviors that are predictive of early onset of psychiatric and addiction risk. We have now replicated in our animal model significant placental changes as a consequence of prenatal THC exposure. And as I showed you before, there are a number of behaviors that we see that last into adulthood. We now also looking in earlier time periods, and we could see changes during juvenile and early adolescence, but we see changes related to motivated behavior. For example, animals have to press a lever once to get a chocolate pellet, and then they have to press 4 times to get the same chocolate pellet, and 20 times, and so on. You see where they work to stop to get that one chocolate pellet. And these adult animals with prenatal THC exposure, they will press a lever 100 times just to get that one chocolate pellet. That could be me. I like chocolate. And they also show greater depression-like behavior, and also that's also evident in other behavioral behaviors that we've studied, such as looking at hedonic state, so that they have this reduced sucrose intake that's also predictive of a more depression-like behavior. So as said, prenatal exposure, we do see have these long-term impacts. So what about the adolescent time period, which is another important area for us, because more teens than ever are still using cannabis, even though nicotine use has decreased. And many studies have shown the impact of adolescent cannabis use, not in everybody, but as I said, there's a vulnerability to certain groups for schizophrenia, or even developing substance use disorder to other drugs, and even developing a cannabis use disorder. So here we use our animal models again, and we can see that adults that had adolescent exposure, they indeed self-administer more heroin. Behavioral traits matter, so it's not that all animals will self-administer heroin in the same way. But we wanted to, again, look at what's some of the neurobiological underpinnings, perhaps, for these effects. And here, for this experiment, we looked in an area of the prefrontal cortex, in terms of that is one of as I mentioned earlier, one of the last regions in the brain to achieve full maturity. And in looking at the cells that are, these are, we can go in and look at the structure of these cortical cells. And we can see that, and it was really interesting, and this is looking at adulthood, for example, that the complexity of these cells differ. So the complexity is these branches, and you can see the video on the left, and you can, the top is a vehicle animal, and the bottom is one that had been exposed to THC during adolescence. And you can see that there's a very big difference to the complexity of the THC exposed adults. One of the things that I wanted to do was to actually know what the molecular signature is in these cells that we saw in the morphological changes. So we actually took laser and picked up each of these cells and then sequenced them to look at thousands and thousands of genes to see whether or not they can give us any molecular insights as to what's maintaining or what's happening to mediate these structural changes that we saw in the cortical cells. The thing that I was actually shocked about at that time is that as compared to normal development, which, there's still a lot of development going on, we could see that those adult animals with adolescent THC exposure, their transcriptome, the gene expression profile in their cortical cells, differed substantially from the normal developing adolescent, late adolescent, early adult brain. They only shared about 5% of the gene that were differentially changed over time. And where they were changed was interesting. They were changed in gene networks related to what we saw on a morphological level, so dendritic sprouting and so on. but also epigenetic mechanisms. And epigenetic mechanisms, so I said we inherit our genetics from our parents, but environment can go in and change how those genes are turned on and off. And these epigenetic modifications were clearly there in these animals, to the point where the genes that regulate the dendritic, the synaptic plasticity, how the synapses talk with each other, in vehicle animals, in normal animals, there's not that much crosstalk between them. But in those animals, adult animals with the adolescent THC exposure, there was a huge reorganization. So we know that adolescent THC alters the structural development of the prefrontal cortex and actually reorganizes the gene networks relevant to epigenetic regulation. So I mentioned in the beginning about the fact that the THC concentrations have really skyrocketed. Today's cannabis is not the normal cannabis. It is, when people talk about GMOs and all of these things, these have been bred now to be very potent with high THC. Just looking at time. Okay, good. So those are some of the pathways that can lead into addiction. And so the question is, how do we get out of it? And I'm going to just end with one particular path that we have been trying, and but there are a few others that I'm happy to talk about in the QA time. And it comes back to our developmental studies, because as I mentioned, when we look at the developmental effects of cannabis in our animal models, we would see that, for example, they self-administered more heroin. But when we talk about that, we're studying cannabis in the animal model, we were actually studying THC. And cannabis is made up of over 140 cannabinoids and many more other chemicals. And we decided to look at another cannabinoid, cannabidiol, which was the second highest cannabinoid, still extremely low in concentration in the cannabis plant. But when we looked at it, we actually saw that it had a different effect on heroin vulnerability. And here we saw that animals, when they're, in contrast to what we saw with THC, these animals, when they were shown a cue, so when animals, and as humans, when they associate the drug taking with something in the environment, and here, for example, like a light in their environment, and when we show them the light, they'll start pressing the lever. They're not getting into heroin, so what we call that heroin seeking. And CBD actually decreased their heroin seeking behavior, and it lasted even weeks after their last CBD administration. Also, one of the things that was important for me in the CBD effects that was interesting is that when we studied the brains of people who have died with a heroin addiction, excuse me, we can see that there's this reorganization of the genes in their striatum. And here it relates a lot to changing glutamatergic system. And we can find the same things in animals that self-administer heroin. And interestingly, CBD normalizes some of those glutamatergic changes. Excuse me. So we decided to look at whether or not CBD, cannabidiol, could be effective in human studies, and carried out double-blinded placebo-controlled studies. And in the same way, we showed people who had a heroin use disorder, they were abstinent, and we showed them different cues and measured, for example, craving. And we could see that when it was unblinded, that those who had received placebo, they craved, and CBD reduced that. We brought them back in the lab a week later after their last CBD administration, and it still reduced their craving. Also, anxiety was very strong in people who were shown the video cues and had received placebo, and CBD reduced that. A week later, it still reduced their anxiety. Here, we also looked at physiological measures of cortisol, and when individuals were shown drug cue, their cortisol levels went up, and CBD reduced that. Their heart rate went up when they had been given placebo and were shown the drug cue as compared to the neutral cue, and CBD reduced that. So for us, cannabidiol holds promise for opiate use disorder, at least for craving and anxiety, and those are things that we're trying to look at in these larger clinical trials, and right now also looking at in vivo neuroimaging in humans. So there is a long way to go in translating a lot of the work that we have done in understanding some of the neurobiological paths that enhance addiction risk. But a lot of the data is really giving us insights into new treatments. And one of them, as I said, potentially cannabidiol. Another one that I didn't talk about, and if I is with the epigenetic changes that we see, that we're able to normalize some of the opioid seeking and taking behavior through modulating these epigenetic changes. So with that, I'm going to thank a lot of people in my team. I mentioned Jackie, Nissa carrying out the prenatal studies with placenta, and Greg with the human placental changes, Alex working on some of the CBD studies as well. And I mentioned Yoko for masochistic studies with looking at, for us going down the path of studying humans longitudinally from prenatal exposure, and her team, and El Salsas, who's an amazing addiction medicine physician who has been a great collaborator in, my really great team with clinical team and Li Wang doing the neuroimaging with CBD, which we hope will get moving more, and Karen Bach, who with the CBD clinical trial, and this is the clinical team as well. So with that, I will take any questions that you might have. Hopefully I left a little time.
But I thank you so much. This was so wonderful, and we just really appreciate your virtual visit and your talk and your meeting with students earlier today. So thank you.
Thank you very much. I enjoyed my visit, actually. I really enjoyed it. Thank you, guys, and thanks again for the invitation.
Thanks
Bye.
a lot,
Thanks
Dr.
to all
Hurd.
the MIND members and friends.